Hi, my name is Caroline Andrews, and today I will be doing my protein project part 2 presentation on my protein, which is KRAS, and its mutation, which is the G12C mutation. And I will be presenting to my Nana, who is not in STEM, and she actually has pancreatic cancer, so I felt that a presentation such as this would be interesting for somebody who could probably relate to the results of such a mutation. So yeah, we'll dive right in. So KRAS is a protein found inside cells. It's mainly in the cytoplasm. So if this was a cell, we'll say this is a cytoplasm, cell membrane, cell membrane. You're going to find your KRAS right here not between, but in the cytoplasm right up against the cell membrane. It works as a control switch, turning off and on signals for cell growth and division. So if it's on, cell growth is promoted. Off, those signals aren't sent, and that's just how the cell regulates growth. KRAS is usually more studied in epithelial cells of the lungs, the pancreas, or the colon, in your case, pancreas, which is why we see mutations in the protein are linked to cancers, again, for you, cancer of the pancreas. The mutation I am focusing on is called G12C, and we'll see a little bit more about that later. So let's just take a quick look at our model. This is going to be your normal KRAS protein. This blue structure is a KRAS. A normal KRAS is going to be more globular, a bit more lumpy, and more compact. We just have things falling. This is going to be what we would call a tetrary structure. So it's the 3D model, it's everything folded together, a more simple model. And then we also have the secondary structure, which would be more representative of your alpha helices and your beta strands. Yes, strands. So looking at the tertiary structure, we have this kind of eye-shaped pocket here. So this is where your molecules like GTP or GDP are going to bind. So we have GTP right there right now. That's going to be the turned on molecule. So it's going to send the signals that promote the cell growth. This is going to be your GDP, and when that's in there, it's going to turn those signals off. So let's compare to the mutation. So looking at a regular protein, we have a white dot here, and in our mutation, we have a red dot. This white dot's your glycine. Notice how it starts with a G. It's at position 12, G12. Glycine's your smallest amino acid and has a very simple side chain. It allows it to fit easily beside the binding pocket. Now, over here, we have our mutated. This is cysteine, C. So G12C. C has taken over 12. That's where we get the name of the mutation. Cysteine is a much larger amino acid, and it contains sulfur. The sulfur slightly changes the shape of the region of the protein, and makes it more chemically reactive and can affect how the protein folds and interacts with nearby molecules, such as your GTP. The mutation's going to occur right there near your GTP binding pocket, and this is going to make the control switch function of the KRAS a little bit wonky. So this is going to interfere with how the protein turns on and off. As a result, the KRAS often stays active longer than it should, sending signals to tell the cell to grow and divide. Over time, these constant growth signals contribute to cancer, hence why the mutation causes cancer. Finally, I just want to touch on some limitations of these models that I do have here. First, the model is much larger and much simpler than a real KRAS protein. The real KRAS protein is made of many, many, many amino acids. Second, the shapes of our helices and strands, including even this one, are much simplified compared to a real protein structure. And then lastly, all the models that I made today are static. They can't move and they don't change shape. So that's not really representing everything, but it's a good introductory, especially for somebody such as yourself who might not know much about STEM. Overall, the model just demonstrates how the structure of a protein relates to its functions and how even a single mutation can disrupt normal cell signaling and contribute to disease. Thank you.